This is a really cool lecture because it touches on a point that is going to come over and over and over in a lot of future lectures. And one of the most important reasons that we need to learn free radicals, oxidative stress, inflammation, and how antioxidants work is they are the free radicals and the inflammation is one of the key underlying drivers of most of our chronic diseases, and so many symptoms and disease onsets are going to be because of oxidative stress. And then we have to have the antioxidants in our body available to be able to fight them. So a little bit of the lingo, we need to remember that when we talk about free radicals, those are going to be molecules that have lost their outer electron and are very highly reactive. And if we recall, they'll react with basically anything that they can steal an electron from. However, the most damaging molecules that free radicals do attack are going to be the DNA backbones, your tertiary protein structures, because it attacks the folding, and that can cause significant damage to the protein. And then if it damages it enough, we can lose that tertiary folding, and then the protein will go into a induced suicide as an apoptosis and recycle itself. And that means the protein, whether it's a channel or a transport or an enzyme, that means it's not there to do its job. And then of course the last one, which is a huge problem, especially in cardiovascular disease, as we will see, and a lot of the metabolic disorders, is going to be that lipid peroxidation, and that's the free radicals attacking the PUFAs, which are your polyunsaturated fatty acids in your plasma membranes and on the outer core of the lipoproteins, which are our lipid transport proteins, which we learn in detail. So having a system of oxidative stress, by definition, is an imbalance in free radicals to antioxidants. We know that the body has a natural generation of free radicals that are normal physiologically generated free radicals. They have a very good purpose in their environment. We learn that they're in the electron transport chain as the molecular oxygen is oxidized with the electron transport to give us the energy molecule ATP. We also know that it's going to be very prevalent as our immune cells attack foreign invaders and microbes. That's how we fight off a lot of the fungus, parasites, microbes, virus. We need that oxidative burst, which is basically the cell using free radicals to damage the microbes. And our body is designed, it's brilliant. We know exactly metabolically and molecularly how to handle all of these things. So in a system of healthy homeostasis, normal amounts of free radicals and normal amounts of antioxidants are perfectly balanced, and we don't have oxidative stress and damage from free radicals. However, once a system is out of balance, we lose homeostasis and we shift into a body habitus of inflammation, oxidative stress, free radicals, and then we have too many of them and not enough antioxidants, because if we excessively increase free radicals and the cytokines and a lot of the inflammatory chemicals, then what happens is the intrinsically available, that basal level of antioxidants, which are designed to balance the free radicals, once those get heightened and occur in higher excessive abnormal aberrant levels, then it uses up our antioxidants. So we're learning how to protect our antioxidants so that they can remain available. And what we have to realize is that just like the free radical has lost its electron and it's reactive, it's going to be quenched and scavenged, meaning the antioxidant, which is going to scavenge, hunt, find it, go attack it, right? Quiet it down is what quenching does. So we scavenge to find it, we quench it to quiet it down, and we do that by donating it back its electrons so it stops being reactive. So when we're talking about antioxidants that are that act by themselves, not in an enzyme system, we are going to call that a solo antioxidant, and those are reducing agents. What is so key important is that those reducing agents, as we're going to learn in detail, those molecules and how they fight free radicals and protect our bodies from damage, aging, and degeneration, is going to be because they have the available electron as a hydrogen atom to donate. So before we dive into the details and mechanism of action of how our primary antioxidants protect us, we have to appreciate that they are reducing agents. And as a reducing agent, we need to know the lingo about redox reactions, because all it is a repetitive, perpetual back and forth of donating the electron, hydrogen atom, losing it, donating it, losing it, donating, losing it. And as you can see in the title, we have to make sure that we are always regenerating our reducing agents, who are the fighters. Those are our antioxidants that have that that electron or the hydrogen atom to donate. So there is going to be some practice lingo. This might look familiar because it was an earlier metacognition. So I really want to focus on the lingo, because we need this to go forward as we move into the antioxidant mechanism of action of a reducing agent through its redox reactions, and why and how the antioxidant reducing agents are going to need to be regenerated in order to sustain their functional ability and physiological role as antioxidants. So when we think about the two molecules, we have the reducing agent, which is going to be our antioxidant, which has the electron and it has that hydrogen atom. And then down here is going to be the oxidizing agent, and that's going to be the free radical, the free radical could be a molecule that reacted, that no longer has that outer electron. So it's now the free radical, as we learned. Very importantly, the top four, there's more than this, but the four we're really focusing on is going to be that superoxide anion. That's the oxygen 2-, our peroxynitrite ONOO-. Very potent nitrogen reactive species. We learn how important that is for vasoconstriction and blood pressure and hypertension. And then, of course, the hydroxyl radical, which is your OH-. And then, of course, we have hydrogen peroxide, which is actually not a radical species. However, it's a very potent oxidative stress molecule. So we're going to learn how our body deals with the hydrogen peroxide. But in the case of the free radicals, those could have also, those are the top free radicals, but in a system, for example, lipid peroxidation, that's going to be where the hydrogen atom on the hydrocarbon chain of a polyunsaturated fatty acid, the free radical, that reactive species, comes by it and it steals its hydrogen. That's going to leave a reactive species on the hydrocarbon fatty acid, because now it's missing it. So we have to think about what, the lingo of how oxidative stress occurs in reducing agents and antioxidants is going to be who gained it, who lost it, who stole it, and who donated it, because we have to determine if it's the oxidizing agent or the reducing agent, and which molecule got oxidized, which one got reduced. And then that's going to be the chaotic lingo of how we have to learn how reducing agents are antioxidants and why we need to have them regenerated. So when a molecule has its electron, has its hydrogen atom, it's reduced. It's also called the reducing agent. If it has the hydrogen atom electron to donate to the reactive species, to the free radical, to the other molecule radical who had its hydrogen stolen or lost it to a reaction, then that's going to, they're going to gain it. And that's going to be, they're going to get reduced. So if you gain the electron, if you have the electron, that's going to be you get reduced. Now it sounds confusing because normally we think of reducing as like lessening, right? So it's kind of confusing, which is why I'm putting a pretty good focus on it. So the reducing agent is going to be the molecule, the antioxidant that has the proton, I keep saying proton, that has the electron or hydrogen atom to donate. However, once it does its job as an antioxidant and it donates the hydrogen atom that it has, right, in the reduced state, it then gets oxidized. So as soon as our antioxidant over here, as our reducing agent, does its job scavenging free radicals and quenching the molecules that are reactive radicals because they don't have the electron or the hydrogen atom, as soon as the antioxidant does that, it donates the electron, it donates the hydrogen atom, and then it no longer has it. Now our antioxidant is oxidized. So an antioxidant reducing agent gets oxidized as it donates it. Okay, so that means it has to get it back. And that's what we're going to learn as the reducing agents, as our antioxidants have to be regenerated, and how all the reducing agents are potential regenerators because they're just going to be around giving back, donating back the hydrogen atom. So the reducing agent has the hydrogen. Once it donates it, it no longer has it. It has been oxidized. So if it has, it also gains. If you gain it, you have been reduced. Okay, so if you lost it and donated it, you got oxidized, because once you no longer have it after you donated it, then of course you're going to be in an oxidized state. And then in order for the antioxidant to be able to do another round of donating, it has to get it back. And we're going to see that that is done by regenerating, by reducing agents that are able to regenerate. So there are different pictures and again, different words. So as the reducing agent loses the electron, and in our case, we're also talking about the hydrogen atom, it becomes oxidized. So this can now go the other way. And once it's oxidized by a reducing agent who can donate the hydrogen atom or the electron, we'll now just get it back. So this is boop, 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 boop. It goes back and forth and back and forth. So we have to be sure that we are watching the lingo. So I'm not going to go over these with you. You can practice these on your own. However, pay attention that the what the answer is and then what the message is, because that's going to explain how the lingo goes. Okay, so this is going to be really good practice, and then we'll do this again. We'll practice it before we go on to the exam. So I'm going to go ahead and pop into the lecture. So the regeneration of antioxidants is vital for them to be able to continue as an antioxidant. That is because antioxidants are reducing agents. Antioxidants have the hydrogen atom that they are able to donate. However, once that reaction happens, they have to be regenerated. If you can't do this continuous cycle of donate, receive, donate, receive, donate, receive, then the antioxidant can't perform another round. So that's going to be a really key concept. So let's look at the two that we've already learned. We just might not have realized that they were part of this whole electron sharing, donating situation. So when we think about the B2 and the B3 that we've already done a focus on when we studied what happens during macronutrient conversion into ATP, right? So when acetyl-CoA, our common molecule of fats, carbs, and proteins, is going to be converted by the body into the energy molecule ATP, the acetyl-CoA will enter the TCA cycle. As the TCA cycle spins through its enzymatic reactions, that acetyl-CoA gets oxidized through the reactions, and as the molecule at the end gets generated, we also get the reducing equivalents. Got to know that. The reducing equivalents are going to be generated from the TCA cycle through the enzymatic conversions and oxidation of the acetyl-CoA from our macronutrients. So the reducing equivalents are NADH and FADH2. Now, those are the B2 riboflavin and B3 niacin reducing agent derivatives. So these are our physiological reducing agents. B2 and B3, in their forms, FAD is oxidized, FADH2 has gained the hydrogens and is reduced. In our niacin derivatives, NAD plus does not have the hydrogen that's oxidized, and it gets reduced. The reduced has gained the hydrogen, and that's now the reducing equivalent, the reducing agent, NADH. Now, the niacin derivative NADP and NADPH is similar. However, we're going to see that NADPH is going to be a very vital reducing agent in our solo antioxidant regenerating system. So we'll put a heavy focus on that going forward. But for this introductory few concepts, be sure we're recognizing that the B2 and B3 are reducing agents that are sharing and transferring electrons. So NAD and FAD with the plus, meaning that they are in their oxidized state, once they go through that redox reaction and the reaction donates, it gives the hydrogen atom, the oxidized reducing agent, B vitamins, they accept, they receive, they gain. All of these are the same concept. They're getting the hydrogen, I feel like a thesaurus, and that's going to be reduced. Now they are reduced. But guess what? It's both, it's bidirectional. As soon as they are in their reduced form, guess what happens? Ooh, the opposite happens. They now donate it back. They lose it. They get oxidized. So it just spins and spins and spins in this like continuous redox reaction, hence the electron transfer, which is what a redox reaction really technically is. So if we look at an example from our TCA cycle, the Krebs cycle, our TCA, succinate with FAD in its oxidized form is going to be converted into fumarate as we reduce our FAD B2 derivative, and now FADH2 is reduced. I am not expecting you to know succinate and fumarate or malate and oxaloacetate. That's not the point. The point is the reaction, that's the redox occurring electron transfer. So don't get lost in the molecules. That's not the focus for us right now, okay? We want to show you that the FAD is our B2 derivative. This is an example from the electron transport chain, and the end result of the reaction is to generate the reduced form of the FADH2 or the reduced form of the NADH, which are then vitally, physiologically right there in the mitochondria, and they're going to go right into the electron transport chain as reducing equivalents. They now go and donate those hydrogen atoms that they just got, and they participate in the electron transfer as molecular oxygen goes through. So that's an example of how we go from oxidized and the functional physiological purpose of the B derivatives, B2 and B3, being reduced, and then how they continue, and then they go right into the electron transport chain to then donate what they just received. So they are oxidized in the original form, and then TCA cycle spins, right, as acetyl-CoA is oxidized through the reactions, generating our reduced forms for the reducing equivalent, NADH, FADH2. Now the reduced form, NADH and FADH2 right here, they are now available, and they're going to go and they donate as reducing agents in their reduced form, their hydrogen atoms as electrons, and they're donating them into the electron transport chain to participate as the molecular oxygen goes through the electron transfers. So we are now going to build on that concept, okay? So we are starting with oxidized being reduced, and now the reduced can now turn around and donate. But as they donated it, now what gets regenerated, now they're back to the original forms. So now NAD here and FAD here, now they're available again to interact with another round of the TCA. So they do this continuous donate, receive, donate, receive, donate, receive. So this is bidirectional redox reactivity of electron potential of electron transfers, and that's how our B2 and B3. So now let's take a look at the NADPH, because this is the B3, this is the niacin derivative reducing agent. And the NADH is also important, and it can do the same concept of reducing and regenerating some of our antioxidants. However, However, specifically the NADPH is a preferred molecule in several of the enzymatic redox reactions. So we're going to put a heavy emphasis on the NADPH. So this is a B3 derivative. Be sure you're recognizing the N is representing the nicotinamide, which has come from niacin. Okay, we want to recognize that not only can it regenerate other reducing agents as a reducing agent, it recycles the other reducing agents by donating the hydrogen back, which then gets oxidized. The other important feature is that this is a special reaction that the NADPH is generated from the pentose phosphate shunt, or there's a couple names for it. I want you to recognize it as the pentose phosphate shunt. So we are going to start with the concept of how it's regenerating. So all antioxidants that have donated their hydrogen have to get a hydrogen back to go do another round. Just like we saw here, where the B2, B3 reducing equivalents are going to receive to donate back to the electron transport chain to be bioavailable again, to interact as their oxidized state to help push the reaction, because the reaction has to use the oxidized state, regenerating the reduced form again to go back into the electron transport chain. So the same thing is happening when we regenerate our antioxidants who have donated their hydrogen. So with NADPH, very, very, very importantly, NADPH is going to donate its hydrogen to recycle oxidized vitamin C, oxidized glutathione, and the oxidized thioredoxin. We'll take a look at these in detail, but this is the first introduction to it. NADPH recycles and regenerates these three vital antioxidants that need to be regenerated so that they can become reduced again and then go do their antioxidant function of donating the hydrogen. So NADPH synthesized from pentose phosphate, B3 derivative, and then it's going to specifically help vitamin C as a sorbate, glutathione, which we learn in a second, and then thioredoxin, which we take a look at as well. Very important molecules. All right, so again, don't focus on the organic acids, right? So we are simply looking at the mechanism of the redox reaction of how we are receiving and donating the hydrogen atoms. So this again is a specific example. You are not expected to memorize this biochemical reaction. We are learning the concept of how the reducing agents are interacting. Okay, so this is a very important reaction. This is called the malate shuttle because we have to shift molecules between the mitochondrial and the cytosol. So what we need to do is use specific reactions that kind of shunt them through. So you'll notice here that the NADP oxidized, okay, oh, did I do that backwards? Let me see. That is supposed to be donating. Okay, so let's make sure that you correct that on your side. I'll correct it on my side, which I just did. Okay, so NADP is the oxidized agent, and it's going this way, and it's receiving. So I was right. Okay, sorry. See, even I have to pay attention to it. The lingo's important. Okay, when it's in its oxidized form, and NADP is going to be receiving, right? It's going to receive the hydrogen and then it becomes reduced. So in this reaction, the oxidized form is going to receive the hydrogen through this reaction, through the enzyme, and then the NADPH has been generated in its reduced form. So now it's in the reducing agent reduced form with the hydrogen because it gained it. So sorry for the typo, but make sure you corrected it on your side. Okay, and then follow along to make sure that you understand that without it, it's receiving. So this is going to be receiving the hydrogen, and it gets reduced in this reaction. So now after this reaction over here, the NADPH, which is your reducing agent, is now available to go interact in another reaction where it needs to donate the hydrogen atom as a reducing agent. So the same molecule, but now when it has to go the other way on the other side of the membrane, our mechanism, the reduced NADH is actually donating. It's acting as a reducing agent where it's giving the hydrogen to the reaction, where the redox reaction then creates and generates the oxidized NAD. So now the NAD plus, which is the oxidized, is going to go react in another chemical reaction that's going to get it back. So it will get regenerated. So, and the cycle just keeps going and going. Again, don't pay attention. The cycle itself is not important. I want you to just get the concept of where the electrons are moving in the redox reaction. So this is another specific one where we're looking at the NADPH. So glutathione is one of the most important intrinsic antioxidants in our physiological system. We make it through de novo synthesis. So our body has the genetic makeup to make sure that we are generating and synthesizing lots of glutathione. And we're going to look at that in just a moment. However, this molecule is very important to have NADPH available in a redox reaction to regenerate it in its reduced form. So you can see here, so take a look down here, both pictures are the same, it's just from different artists. The glutathione, once it donates the reduced, it interacts in this molecule. We'll learn that in a second. I don't expect you to know that yet, so just follow the NADPH right now. That's what I want you to focus on, okay? The reduced glutathione is going to do its job as reduced, meaning it's going to donate its hydrogen. It's going to participate in the redox reaction. Once it participates in this enzyme, which we are going to be learning shortly, it gets oxidized. In order for glutathione to participate in another round, the oxidized form has to be regenerated. So as soon as the oxidized glutathione, which is the end product of it donating, right? Once it donates it, it gets oxidized. So this shows you down here that the oxidized glutathione is depending on both NADPH, right? Reduced NADPH, which is going to react with an enzyme that contains that FAD. So those are going to interact, and that's actually a very, very, very common physiological trade-off redox reaction between B2 and B3, which is one of the very important reasons that those two essential B vitamins have to be continuously taken into the body to make sure that they're bioavailable for all of these redox reactions. So the NADPH is vitally important to regenerate oxidized glutathione back into the reduced glutathione. So NADPH donated its hydrogen atom to glutathione, and it regenerated reduced glutathione, which can now participate in another antioxidant reaction, while the NADPH got oxidized. So now NADP plus is what's left, and now that's going to go react in another redox reaction and reform the reduced agent. So the other key point is that this pass off of NADPH interacting with the oxidized FAD is going to be a very common physiological reaction. And if you look back here, this is what I have demonstrated. The NADP here reduced is going to have the hydrogen. It's going to interact with the enzyme that contains the FAD in the oxidized form. And then as the hydrogen is going to pass this way. Now, in this case, I showed you that it's being put onto FAD as NADP got oxidized. However, what you're seeing here is in how the body works is the FAD is kind of that electron transfer. It's just a pass off. So it's facilitating that transfer instead of keeping it for itself. This is how we're reducing and regenerating glutathione. So key points. That NADPH is vitally important to regenerate oxidized glutathione into reduced glutathione, and it does it by passing a hydrogen atom through an FAD, which is your B2, and then it regenerates the antioxidant reduced glutathione. And then here's just another picture where it's showing the pass off. Okay, so we'll look at the glutathione peroxidase going forward. For the moment, just pay attention to the NADPH, which is interacting to recycle as it donates the hydrogen atom and regenerates our reduced glutathione. Okay, so here is another picture. We are going to pass the hydrogen onto the oxidized form, regenerating the reduced form. So as I had explained, this mechanism of transfer, of donating the hydrogen from the reducing agent is vitally important. So we started with NADPH as the initial solo antioxidant, and we're going to find that each of them, all of the solo antioxidants, which are reducing agents, they all help each other. So we're going to see how, as long as they're available with their hydrogen atom, they can also interact to give it back to one of their other buddies, the reducing agent antioxidants, as a handoff to be like, yo, dude, I got you. Here you go. Go do your job, right? So it's just this back and forth, and that's really cool. That's recycling and regenerating of the antioxidants. So the catch-22, of course, is that every time an antioxidant does its job, it gets oxidized. So the catch-22 is every time the antioxidant does its job, it has to get regenerated, which another local reducing agent comes along and does to give it back the hydrogen so that it can be reduced again, so that it can go donate it to the free radical and quench our free radicals as they get scavenged. So what we're going to see here are these 6, now 5, since we just focused on NADPH, are going to be the primary solo antioxidant reducing agents that we're going to focus on in our physiological system. Vitamin C, vitamin E are essential vitamins. So vitamin C is our water soluble ascorbic acid, depending on what state it's in. The vitamin E is tocopherol, very important. We're going to learn where that is. Glutathione is a vitally important molecule. Again, our body makes it. We can also ensure that we are eating the foods or taking the proteins that make it. So we'll learn that in a minute. CoQ10, yes, that's the exact same molecule that's in the electron transport chain. We'll see that. Alpha lipoic acid, one of my all-time favorite molecules. This is in my cupboard at all times. And we learn a lot more about alpha lipoic acid. However, it is a potent reducing agent with the ability to participate in many redox reactions, which works to our advantage. We should keep high levels in it, well, minorly high physiological doses. And then of course the NADPH, as we had just learned. So once our solo reducing antioxidants, the 6th we are learning here, once it donates its hydrogen atom, it gets oxidized because it had donated it, it quenched it, right? So then it has to be regenerated by another focal or local reducing agent, which can then turn around, and these can all donate to each other in different systems. So there are certain ones that prefer each other, and we'll learn those. However, the overall concept is our solo antioxidants quench free radicals and go scavenge free radicals, and they quiet down and deal with and prevent or fight oxidative stress. However, they too need to be reduced, and they also act as reducing agents to recycle and regenerate themselves and their category of solo antioxidants. Very, very, very important. So let's take a look at vitamin C. Vitamin C, again, is ascorbic acid or ascorbate. It's one of the most potent water soluble. It scavenges free radicals, but it also regenerates other antioxidants, which is important because it's doing both. And then It also regenerates glutathione and vitamin E as the top ones. Okay, so vitamin C is going to be the most important reducing agent to regenerate vitamin E. Vitamin C in the physiological system makes sure that once vitamin E is tocopherol does its antioxidant role and donates its hydrogen, it has to be immediately regenerated and it has to get it back. If you are deficient in vitamin C, which is a water-soluble vitamin, which you have to be taking in every day, or you will eventually, you'll urinate it or sweat it out because it's water-soluble. We don't store a lot of the vitamin C. What's physiologically available is what we really need to deal with. So you have to work at keeping vitamin C in your diet, and squeeze a lemon into your water and cook with lemon, cook with lime, use your citrus. I supplement with vitamin C almost daily. It's delicious. I like it in the emergency packets. It comes with the water-soluble B vitamins. There's also effervescence of vitamin C that come with zinc, which is really good for the immune system as well. So vitamin C, I want you to definitely keep in mind that it's the #1 antioxidant that keeps vitamin E regenerated. Secondarily, as you can see in this reaction here, the vitamin C is helping donate the hydrogen atom and also recycling the glutathione. So glutathione, through the glutathione peroxidase and glutathione reductase, as we learned with NADPH, is going to rely on that reaction of the NADPH with the FAD, which is your B2. So B2, B3 are interacting to recycle glutathione. And here we just saw a reaction where vitamin C does as well. So that's actually really important. Now, vitamin C can be regenerated by NADPH. One of the most important intracellular vitamin C recyclers is going to be the NADPH. And if you run low on that, then you can actually lose your vitamin C, because it will go into a radical and then that will get degraded, and then it's just physiologically inert. So if you can keep it recycled, your vitamin C will just stay and stay and stay functional, because it just keeps changing redox states. But if it does not get regenerated, then it will get completely catabolically broken down and we will lose it. So definitely NADPH. Now, it's a reversible reaction. So we can also see here that glutathione is also going to regenerate the vitamin C. Thiolredoxin is an important molecule as well for vitamin C, and then alpha lipoic acid we will see as well. So vitamin C is going to scavenge the free radicals, and that it will prevent it. Again, as long as we have sufficient antioxidants, the free radicals do not cause oxidative stress. They are physiologically available in our system, and they are scavenged and quenched by free radicals, by antioxidants, as long as we have them. And in order to have sufficient amounts of antioxidants to do their job, we have to have sufficient amount of the reducing agents, which are the other antioxidants. Okay, so let's see. Vitamin E, glutathione, very important. And they will react and react. So you'll see that they recycle and regenerate each other. There's a couple other cool pictures. So the key concept here is that the redox reaction of our antioxidants, well, we're going to learn the sulfur cycle in just a second. The ascorbate, which is our vitamin C, here's a picture, and they chose to use the NAD, however, it's much more commonly NADPH. So it will be able to be both, because they're both reducing agents. And then as vitamin C is going to donate, it also loses. So you can see here, dihydro is going to, that's your reduced state, and then it's just this pass off, and that's how they work in the physiological system. So here are some examples of the regeneration. So we've got the NADPH, which is your B3. We have our alpha lipoic acid, and we have our glutathione. And then the picture that was not from here was the thioredoxin, which also is a potent regenerator of vitamin C. So what the heck is thioredoxin, right? And what about glutathione? These are organosulfurs, and they are categorized in a molecule category called thiol. So be sure you learn that thiol is going to be a molecule that contains the sulfur group. Very, very, very important. We will learn sulfur metabolism in another biochem class. So right now, I want you to focus on these 4 molecules, and these are some of the most important physiological thiols because these contain sulfur. So your cysteine is going to be one of the primary sulfur-containing amino acids. There are others. Right now, I want you to know cysteine. Okay, we'll learn more about that, but right now I want you to learn cysteine is your sulfur amino acid, and that is your thiol. Okay, glutathione is going to also be a tripeptide. It's made up of 3 amino acids. Guess what amino acid is going to be in glutathione? Cysteine. That's why glutathione is a thiol itself, okay, because it contains the sulfur and it contains the cysteine. Alpha lipoic acid is going to be a wonderful molecule, and that also contains 2 sulfurs actually, and that's going to be important in its redox reactivity. And then we also have a thioredoxin, and this is also going to be a cysteine enzyme. So the thioredoxin, the glutathione, the cysteine are all sulfur, and then the alpha lipoic acid is the disulfide thiol. Okay, so when we are looking at the thiols, we can, NAC is going to be N-acetylcysteine. It's a highly bioavailable, absorbable form of the cysteine, which we'll see in a second. So the next concept that you absolutely have to know, and this comes up over and over, so plant this in your long-term memory and potentiation. Glutathione is the tripeptide. It's cysteine, glycine, and glutamate. Cysteine, glycine, glutamate. Cysteine, glycine, glutamate. Those are our thiols because cysteine is the sulfur amino acid, which makes it a thiol. And then because glutathione is a tripeptide of cysteine, glycine, and glutamate, it too is a thiol. So let's take a little bit more look. I want you to know that NAC, N-acetylcysteine, is a very, very potent, wonderful antioxidant that we can supplement with. Not only does it help it keeps your cysteine pool high when you supplement with it. It has been shown to immediately increase the de novo synthesis of glutathione because it's a precursor. Know the term precursor. A precursor is when the molecules are used to make the end product. So cysteine, glycine, and glutamate are the 3 precursors to synthesize glutathione. N-acetylcysteine is a slightly different form of cysteine, highly absorbable and highly bioavailable. And NAC is going to be something we supplement with that can turn on the synthesis of glutathione. We will learn that the NAC, the glutathione, and the cysteine are all also vitally important in the liver detox of chemicals, pollutants, any foreign substances, and xenobiotics. So these come into really, really, really important physiological roles because of their sulfur group, okay? So NAC is also one of those supplements that I keep in my cupboard almost at all times. This is a 600 milligram, and this has a synergistic selenomethionine and molybdenum because of the interactions in the biochemical pathways. So not right now, but in the very near future in another class, you will need to know how the cysteine interacts with the selenomethionine and molybdenum. For right now, I just want you to plant it into your memory that cysteine is the thiol because of the sulfur group, and that it can also come in the form of NAC, and it's your precursor for your glutathione. So here's a cool article from PubMed, and here is the front page of it. So this is the actual article. I thought this was cool, getting a knack for NAC, right? little pun on words, it's cute. Say, through its metabolic contribution to glutathione production, cysteine participates in the general antioxidant activities of the body, quote unquote. Absolutely phenomenal. Keep it in your cupboard at all times. All right, so why is glutathione so awesome? Because it is. It's totally awesome. It is going to be your thiol. It's a tripeptide. It's solo antioxidant, does the donating of a hydrogen atom that it has when it's reduced to fight free radicals directly. It also is brought into the very important antioxidant enzyme system that's going to interact with free radicals through a conversion, which we will be learning shortly. So glutathione by itself is a solo antioxidant, and glutathione participates in enzymatic reactions also to fight free radicals and to decrease oxidative stress, and we will learn what that's going to be doing. So this is important to learn these abbreviations. Okay, these abbreviations are vital, and you're going to see them in medical literature. You have to be able to read them so you understand what the articles are telling you, and I will use them so that you will almost never see me write reduced glutathione. You are only going to see GSH, and you have to know that that is the abbreviation for reduced glutathione. And if you recall reduced glutathione, when we look back at this picture, reduced glutathione is going to be the only form that can function as the antioxidant enzyme. Once it gets oxidized, if we cannot regenerate it back, this cannot participate in the enzyme action again. So here we have GSH, which is the reduced glutathione, and then once it gets oxidized after it does its antioxidant role, we are now abbreviating it as GSSG. That is the designation for the oxidized GSSG. And as you can see, once it's oxidized, it has lost its antioxidant potential until a reducing agent, typically going to be that NADPH, but we can also see vitamin C, that's going to reduce it back. So the glutathione is going to also participate in this enzyme here. So reduced glutathione, only reduced glutathione can participate in this glutathione peroxidase, which we abbreviate GPX. So go ahead and make sure that you recognize the abbreviation, and that is the glutathione peroxidase, and that is going to require the two of the reduced glutathiones. We see that in a few more slides. And then this is going to be a selenium dependent enzyme. So right now we're introducing glutathione. We learn this enzyme system next, so those details will come forward. So for the moment, I want you just to get used to the abbreviations. So one more time: reduced glutathione, GSH; oxidized glutathione, no antioxidant activity, no hydrogen atom, that's GSSG, which is why it's oxidized. And then your glutathione peroxidase is your GPX, okay? So here is the tripeptide. Now, this is cool. Glutathione is so important as acting as an antioxidant that we can actually measure the oxidative stress or the redox potential of the cellular states or the physiological body by measuring the ratio of reduced GSH to the oxidized GSSG. Because if we drop below an optimal level of 10, if we're below 10 to 1 of a ratio, that's going to demonstrate a significant oxidative stress state. That means glutathione is not getting reduced. That means we don't have enough reducing agents, and we are low in antioxidants, and we are high in oxidative stress because it's not getting regenerated back. We do not have protection against many free radicals if we cannot regenerate and recycle glutathione. So an oxidative stress state is going to be associated with a low ratio of the reduced to the oxidized, and a healthy system has a high ratio, meaning that there's a lot of reduced glutathione so that it can continue to participate. That also tells us that there are plenty of antioxidants and reducing agents that are doing their job recycling it. So that is important to understand that that is a recycling agent, and it is the most abundant thiol. So that's pretty cool. All right, so now let's take a little bit of a deeper look at this. So as a reducing agent, it itself can react with free radicals, or it can donate its hydrogen atom to other antioxidants to regenerate them. The other one that we learned is we have to have glutathione peroxidase. This is a selenium-dependent antioxidant enzyme, and then that's going to convert your hydrogen peroxide to water. Very important. We learn that in detail going forward, so add that to the memory base. Selenium-dependent glutathione peroxidase requires 2 reduced glutathiones in a selenium-dependent antioxidant enzyme reaction, and that is going to take that free radical hydrogen peroxide and convert it to water. Very, very important. So as you can see here, selenium is important in that glutathione peroxidase. And what did we say about NAC? N-acetylcysteine is going to be a precursor that's going to support the synthesis as a precursor for glutathione. And then we just learned that the selenium is going to participate in the glutathione peroxidase reaction to fight hydrogen peroxide by converting it to water. So the synergistic mix that Now Foods does, because they're like brilliant, they are going to deliver NAC with the synergistic selenomethionine, which is a selenium bound to a methionine. Now, you don't have to know this yet, not even for this exam. However, guess what? Methionine is the essential amino acid that is your sulfur amino acid that will be converted enzymatically and catabolically into cysteine. Pretty cool. So selenomethionine is a really great choice to stick it into the NAC product. And just a little tidbit, because we'll be doing it next week, molybdenum participates in sulfur metabolism, and molybdenum helps the degradation catabolic process of your cysteine and methionine and taurine to be catabolically broken down to generate sulfur. So that is sulfur metabolism, so important. All right, so let's see. We take a look at that. We've already looked at this, so this should be a review now. Your reduced glutathione has to, with 2 of them, participates with our glutathione peroxidase. That's a selenium-dependent enzyme that's going to convert your hydrogen peroxide to water through an enzymatic conversion. As the reduced glutathione participates in the reaction with selenium, and donates its hydrogen atoms and generates water from hydrogen peroxide by conversion, you get an oxidized glutathione. And then for this to occur again, NADPH with an FAD in the enzyme glutathione reductase, it's going to then regenerate our reduced glutathione. So that is now the big picture of how we are using all of these concepts together in continual reducing agents, regenerating antioxidants, and fighting free radicals. All right, so let's look at the thiols, thioredoxin and alpha lipoic acid. So these also have sulfur. So these fall under the category of a thiol, meaning they contain sulfur, and that's particularly where their redox reactivity is coming from. So we are going to see that the thioredoxin scavenges free radicals, and it also regenerates vitamin C. So the NADPH, okay, it's going to react through this sulfur transfer, and then we're going to get the NADPH reduced, right, thioredoxin. Then it's going to donate it, and then that, as it donates its hydrogen from the thioredoxin reaction, it will then donate it to the protein, or they're using a protein here, or to vitamin C in our regenerating case. So it's going to pass it off, and then we recycle the thioredoxin. So it gains and loses its hydrogen atom, and just make sure you recognize that it is the thiol with the sulfur group, and it's actually a cysteine that it has. So this is, again, another example where the thioredoxin enzyme systems, when we're looking at how our bodies are using these redox reactions, what I want you to focus on is, number one, as I just said, thio, meaning sulfur containing. The thiol that this enzyme and protein has is going to be the cysteine sulfur amino acid, okay? And then what we're doing here is the FAD is going to be your B2. And as you can see, the oxidized FAD, as we've already learned, is interacting with the reduced B3, and this is a common reaction. FAD is going to then participate in the regenerating and recycling of the thiol. So as FAD does its job, it's then depending on NADPH as its reducing agent to come and recycle it. So this is a beautiful example of how the redox, one just donates to the other, one just donates to the other, and it's this flux of just electron flow of regenerating our oxidized and reduced molecules. So that's how you are going to read that. It's an example of the pass-off. You are not responsible for the different forms. This is just a pretty picture from the Linus Pauling Institute. Wonderful, wonderful resource. I want you to know that alpha lipoic acid is abbreviated ALA, and that's your thiol containing 2 thiol sulfur groups, okay? That's 2 sulfur thiol groups. And the reduced alpha lipoic acid, ALA, is your dihydro, 2 hydrogens, dihydro, 2 hydrogens. So that's how it's going to be able to interact in the disulfide, 2 sulfur groups with 2 hydrogens. So this is an example where alpha lipoic acid is fighting the hydroxyl radical. So be sure you recognize, again, OH minus is your hydroxyl radical. And here's a pretty picture. Now this is cool, okay? So don't get too confused in here. I'm not gonna make you draw this picture, but this, you have to get the concept. This is an example where our alpha lipoic acid, right? Here's our alpha lipoic acid. Because of its redox potential, it's interacting on one side to be regenerated by NADPH as it goes from reduced to oxidized. In its reduced state, it is now regenerating CoQ10, it's regenerating vitamin C, and it's regenerating oxidized glutathione. Alpha lipoic acid in its dihydro state is in this picture relying on NADPH to be regenerated so that it can react with all of these. Any of these 3, this is oxidized CoQ10, this is oxidized vitamin C, and this is oxidized glutathione. All of those can be regenerated if dihydrolipoic acid is in the vicinity to donate back its hydrogen. So this is a beautiful picture of how they're just all sharing hands. They're all best buds. They're all like, hey, I got you. All right, cool. You got it? Yeah, I got it. You need it? Yeah, I need it, right? It's just kind of like a telephone game. It's how I picture it in the body. So then this is also on this side, this is something we haven't learned yet, but this is a beautiful picture where dihydro alpha lipoic acid, or as we're reduced alpha lipoic acid, reduced ALA, is going to make sure that CoQ10, vitamin C, and reduced glutathione are all in their reduced states. And then all 3 of these can help recycle our vitamin E. You haven't learned this picture yet, but this is a demonstration of CoQ10, vitamin C, or glutathione in their reduced states, all being able to recycle that tocopherol radical. This is vitamin E if it's not reduced, and then it has to be regenerated to be a functioning antioxidant again. So alpha lipoic acid is really important. All right, so tocopherol, vitamin E, it is one of the most important plasma membrane antioxidants. Let's think about why, okay? Why is vitamin E is fat soluble? And then if we look at a phospholipid, it's going to nudge itself into the plasma membrane and kind of float along next to all of the fatty acids that are part of the phospholipid and the structure. So what did we say about the PUFAs? So the second one on these, okay, the plasma membrane is made up of your PUFAs, polyunsaturated fatty acids. What's highly susceptible to oxidation and oxidative stress? your PUFAs. They're hydrocarbon chains. That hydrocarbon chains means that those hydrogens are going to be stolen by any free radical that wants a electron or wants a hydrogen. So PUFAs in plasma membranes, every single cell has a plasma membrane, and then our lipoproteins are made up of a phospholipid outer core. Those are all PUFAs. So we have to make sure that we have vitamin E, our fat-soluble vitamin, nudged periodically into our plasma membranes and lipoproteins. Now, this is cool. You have to know this. Vitamin E participates in 2,000 adjacent PUFA protective vitamin antioxidant redox reactions. One vitamin E in the plasma membrane can protect 2,000 phospholipids in a chain. That's phenomenal. That's why vitamin E is really important. But guess what? If we do not recycle vitamin E after it's done its antioxidant job, it itself, being in the position, it's also a radical. So if you cannot regenerate vitamin E, we're going to have oxidative stress caused by vitamin E radical. The vitamin E radical is what vitamin E is after it does its antioxidant job by donating its hydrogen. So we call that oxidative stress of the PUFAs and of the lipids, lipid peroxidation. So what vitamin E, as tocopherol, is doing in the plasma membrane is preventing lipid peroxidation. Vitamin E prevents lipid peroxidation. If vitamin E is not regenerated by vitamin C, CoQ10, alpha lipoic acid, glutathione, if all of those, NADPH, if all of those reducing agents don't recycle each other to make sure that they're bioavailable to recycle and regenerate tocopherol vitamin E, then vitamin E itself causes lipid peroxidation. That's why vitamin C is being the most potent antioxidant to regenerate vitamin E is super important to make sure that we have plenty in our system. So if we have oxidative stress and it's soaking up and using up all of our antioxidants for erroneous, excessive body habitus, inflammation, and other sources of oxidative stress, then it can use it up, and then our vitamin C is going to not be available to protect our vitamin E, and then we can have a system of lipid peroxidation, which is going to be very, very, very bad. So this is another picture demonstrating how the vitamin E can be, it gets oxidized, and it will depend on the other reactions so that our vitamin C, glutathione, and all of our other reducing agents that are all reducing each other are bioavailable. Another key word that I have to show you here, so plant this in your memory as well with the lipid peroxidation. Because vitamin E prevents lipid peroxidation of the 2,000 adjacent PUFAs, we call vitamin E a chain-breaking antioxidant for the plasma membranes. So the propagation, because think about all of those hydrocarbon chains. If vitamin E can prevent the oxidation of one fatty acid hydrocarbon chain in these fatty acids of the plasma membrane, every second position is a PUFA. However, both of those are fatty acids, and they're both susceptible to oxidative stress because they're long hydrocarbon chains. Any of these could have their hydrogen stolen. So vitamin E comes in and protects 2,000 of them from having their hydrogen stolen. And that's called preventing lipid peroxidation. And when vitamin E does that, that's called chain breaking. Vitamin E is one of the most important chain breaking antioxidants to prevent against lipid peroxidation. We will find that going forward, that's largely vitally important for cardiovascular health, and we learn that in detail as well. So lipid peroxidation, I just explained that as well when we look at the plasma membranes. So here's a beautiful picture to bring the concepts together. The concept is they are all keeping each other reduced. You need one to reduce the other so that can reduce the other. So we've kind of moved. Yes, they're still fighting free radicals. However, they have to depend on regeneration for the antioxidants to all sustain. So number one, when we are thinking about essential vitamins, B2, B3, vitamin C, vitamin E, those are vitally important that we have to ingest every day and actively, proactively make sure that they are in our system, in our diet, and being absorbed so that they can participate in all of these different reactions. And then they are all regenerating each other. So you're going to see here, this NADPH is vitally important. It's always there doing one of the regenerations of the other. Our thiol cycle is going to be any of our sulfurs. So what do we see here? We see our alpha lipoic acid, the thioredoxin, or the glutathione. So this is, the thiol is simply explaining that that's any of those three, which could participate here because they all do. And then those are interacting with that vitamin C and they're getting regenerated. And then the vitamin C, again, is almost always the vitally important regenerator. However, the others, they can participate. And we're going to learn in a minute, we're going to add something to our picture. I'll jump ahead here. CoQ10 can also not only regenerate vitamin E in the plasma membranes, but CoQ10 can also participate in some of the prevention of lipid peroxidation and protect the PUFAs and the plasma membranes. So what is CoQ10? It is ubiquinol. You are familiar with CoQ10 because of the electron transport chain, and we're going to see that it is very involved in the electron transport chain. So when somebody is low in CoQ10, they can actually experience not only significant fatigue because of the role in the electron transport chain, but it can also cause significant loss of protection for that lipid peroxidation, assisting vitamin E in protecting plasma membranes and lipoproteins. So what we need to remember here is any person on a statin anti-cholesterol drug is going to inhibit the body's natural ability to synthesize CoQ10. It comes from the same pathway as the cholesterol synthesis, and that's what the statins do, is they inhibit the HMG-CoA enzymatic reaction, and that's going to prevent the intrinsic de novo synthesis of CoQ10. So anybody on a statin is going to have deficient CoQ10 because their body can't make it. One of the clinical pearl here is somebody with leg cramps. Is very common to have like the myalgia and leg pains because of the statins inhibiting their CoQ10. So CoQ10, this is interesting. We'll see the electron transport chain in just a second. Its role in the electron transfer, in its physiological role of the electron transport chain, is one way that CoQ10 is regenerated to be bioavailable as a reducing agent, as an antioxidant. I think the body is so cool. It just like knows what to do, right? And then we also can see that CoQ10 and alpha lipoic acid, they're like best buds, right? So glutathione and like vitamin C and vitamin E, like they're like one click over here. They're like total best friends. And then on the other side, CoQ10 and alpha lipoic acid, they're like always helping each other. And then we'll learn that one more key one that significantly can improve fatigue and energy levels is a combination that you can supplement with is Alcar, which is L-acetyl-carnitine. This is not on your exam. This is just a clinical pearl. We'll learn this in detail later. But alpha lipoic acid with CoQ10 with Alcar, which is acetyl-L-carnitine. You can't. Fatigue doesn't stand a chance. It helps metabolism. It helps energy. So they're the best buds. All right. So here's a cool picture demonstrating. Again, this is a Linus Pauling of Micronutrient Institute. And we are looking here for the CoQ10 interaction with vitamin E. We already took a look here. So what I want you to know here is that the CoQ10 is involved in 3 of the 4 proteins in the electron transfer. You don't have to draw this. You don't have to memorize this. I want you to just notice the picture that it's involved in 3 of the 4 complexes in order for CoQ10 to be able to participate in the molecular oxidation generating ATP from the electron transport chain. By now you should recognize this iron sulfur complex, which we learned is a redox state of iron. And then over here we've got our cytochrome C, which is the heme iron with copper, which we learned in our biochem. So the another picture of the electron transport chain and the CoQ10. All I want you to recognize is that the CoQ10 is in 3 of the 4 reactions for protein electron transfers of the 4 protein electron transport chain. And I'm not going to test you on this, but these are some of my favorite products that correlate with the antioxidant reducing agent and ensuring that we have enough of them. All right, I hope you enjoy the antioxidants as much as I do and have a much better understanding of why they are so vitally important and how they function in our body.